going to actually start with the acknowledgements, because what I intend to do requires a multitude of collaborators, because that is the only way you can reach across this many orders of magnitude of complexity. And specifically, I want to parse them in terms of their involvement. Everything you're going to see today is a collaboration with Neil Hunter over the past decade. And his lab provided not only structural data in the form of AFM, crystallography, NMR, and component information such as mass spectrometry and so on, but also the testing of the kinetics of the system as a quality control of the structural models as they're converted to functional models. Now, I want to particularly give tribute to my late mentor, Klaus Schulten, who provided the ecology upon which such work was made possible. And in the audience, I would like to acknowledge my friends Anna and Torsten, as well as, of course, Zan. Anna Damjanovic and Torsten Ritz worked with Klaus Schulten long before I did on some of the key proteins in purple bacteria. So the approach today, I'm going to wear 3 hats. Hat number 1, puzzle solver. I need to build structural models on how these bioenergetic domains are assembled. Then I need to be a physicist and ask myself, given the structural information, this bit of structural information, what are the functional consequences? These are tested against experimental outcomes in the same way that experimental input was used in the building of the structural models. The third hat I want to wear is that of an economist for a cell, if you consider ATP as a currency. Because if you do so, you can test cell-scale observables with this approach. This is going to be my final aim. So this, as I said, is an effort that was a decade long with Neil Hunter's lab. So from the beginnings of a few proteins at a time, this starts to climb the scales with increasing multi-scale concessions to detail, which ends over the past year or two with tests against cell growth experiments as a function of illumination conditions. As I said, this cannot be done with one method only. For example, molecular dynamics will never reach down to electronic level details. You have to do that with quantum mechanical language using effective Hamiltonian descriptions, and it will never reach up far enough to test against cell scale complexity. So you have to basically change horses each time, where the output of one approach becomes the input of the next, and so on. So if I want to motivate you, I want to answer one question today. Just how good is a bacterium as an energy harvesting device? Such a question has to be quantified, and I intend to do so specifically in terms of energy return on investment. If you were to buy a device engineered by a human, this would be a quantitative measure of how good that device is. So this would be the ratio of the energy produced over the lifetime of this device divided by the cost of manufacturing this device. And so this uses ATP basically as a currency in the cell. So I'll talk about 3 separate structural models, that of purple bacteria, cyanobacteria, and plant grana. You'll see that there are different levels of complexity involved in this, and I'll not be able to go as far with cyanobacteria and plants as I can with purple bacteria. But the roadmap then is build structural data. Start from structural data, build structural models, iterate this with your collaborators until you have a consensus about what the structural models, what the structural organization is, and then build functional models in terms of the kinetic implications of the structure, and then test them against cell scale behavior. And there's a corresponding temporal hierarchy from femtoseconds that slowly starts climbing towards seconds, and there's a structural hierarchy as you go from atomic detail to protein, organelle, and cell level detail. And with purple bacteria, we can go all the distance, whereas the work in cyanobacteria and plants is currently ongoing, and it's probably a couple of years of additional work to reach the same depth. So the structure implies function, and the function in purple bacteria versus cyanobacteria and grana have a lot of overlapping components, and in fact they have overlapping components also that correspond to respiration, as my colleague Rafael Barnardi will talk about tomorrow. And luckily, I have the good fortune of talking after Greg and other colleagues, so I don't need to introduce a lot of these structures at the moment. So I want to give you a glimpse about how the structure models are built. So instead of doing it for all three of these, which will take the rest of this talk, I will instead show only one. So a plant granule domain, how do you start with data, how do you end up with a structural model? It's basically a puzzle solving and pattern matching problem. So raw data gives you something about the protrusion profiles of the membranes, and you know the building blocks from, let's say, crystallography. And then you apply image feature detection protocols, so that is to say a couple of thousand lines of code to match patterns to the protrusion from the membrane as you get from the data to produce a map of where the visible parts of the individual proteins are. So this is not enough because AFM doesn't tell you where, for example, the LHC2 units or the rest of the PS2 super complex subunits are. So these are mapped basically by eliminating the constraints of the super complex organization and then basically painting the remaining holes with the LHC2 trimers. And you know the component list also from mass spectrometry. So in short, what you see is an atomic detail structural model. However, it is blind in terms of its accuracy to the limitations of AFM as a method. So this has been done for the other models. I'll not tell you how the purple bacterial model was constructed, but this structural model has been around for a while. You might have seen it in the cover of Bob's new edition of Molecular Mechanics of Photosynthesis. Side note, the first edition was actually one of Thorsten's images. So we are keeping it in the family. And this organization, this is essentially tomogram data from Lena Korkutas at Cornell, who initially worked with Wolfgang Baumeister. You see actually a fairly dense pecking pattern of chromatophores in a purple bacterium. And similarly, you can play the same structural construction game with cyanobacteria, in which case there are domain segregations of proteins, and sometimes this pattern actually depends on the growth conditions. So you basically get a gallery of membrane arrangements on the key proteins. One funny side note here, Prochlorococcus, I learned, is actually the most abundant photosynthetic species on the planet by mass. So this pattern very likely is the most abundant light harvesting pattern on Earth if you weigh it by species. So what do you learn from this? Basically something about structural organization, but more importantly, you can begin to ask questions about how the functional implications follow from structure. In the beginning, we have to begin from the quantum mechanical interaction of electronic states and the migration of energy, which later on harvested in a various in a sequence of steps towards ATP synthesis. So that means that I need to climb the scales from individual pigment to protein to organelle to cell scales, which requires a multitude of methods. And the first step in this is basically a description in the form of electronic excitation transfer in the language of effective Hamiltonians. For purple bacteria, luckily, you can get away with using modified Förster theory because there is a convenient separation of pigment clusters from one another, and the electronic states and the energy transfer rates between them is fairly... Pardon me, there's a question? Yes? No? Okay. The transfer of excitations between pigment clusters is actually fairly well explained by modified Förster theory. And if you iterate such a computation through, let's say, hierarchy equations of motion formalism, the bulk observables, such as excitation lifetime or quantum yield, actually do not change much. You can repeat this for any of the structural patterns you have seen, so you can begin to ask what are the bulk implications in terms of, let's say, optimization criteria of the individual protein complexes involved. From this step, you want to climb towards the rate kinetics of ATP synthesis, where electronic excitation kinetics is an input parameter only. And if you apply molecular dynamics as a window, that gives you access to sort of a microsecond time scale. From this kind of an approach, it is a simulation that is performed by Abis Singaroy as well as some of the colleagues at Urbana-Champaign, based on structures we published over the past 4 years. And molecular dynamic simulations give you basically access to some key rate limiting parameters, which are essentially diffusion elements. So you learn about electrostatics behavior, which gives you the ability to coarse grain the Brownian dynamics. And these are some of the first rate limiting turnover parameters in the system prior to ATP synthesis. The rate kinetics of ATP synthesis, I will not get into any of the math in here, but essentially the cycling time of quinones between reaction centers and the BC1 complex is one of the key rate limiting components. And in terms of the stoichiometry, the BC1 complexes are seen to be actually a major rate limiting element. And if you do an expansion in terms of the leading order terms of rate kinetics, this permits you, without having to pay the price of a computation of the entire chromatophore over the length scales and time scales involved here, you get an estimate of the ATP synthesis rate as a function of chromatophore composition and illumination condition. And this also gives you access to the efficiency of energy conversion from photon capture to the production of the final photoproducts. And so quantum yields, as you know, typically clock at about 80 to 90%, whereas energy conversion efficiency at this point has, of course, dropped to something like 10% or less. And like it follows naturally that the chromatophore is optimized for low light. I mean, we know that purple bacteria live in the bottoms of ponds with not much light access, but so this is something that is not put in by hand into the computation. It basically follows as a quality test that the optimization regions are actually consistent with what you know as a physiological preference of purple bacteria. You get to test what aspects are selected for in the composition of a chromatophore. The first lesson is that the chromatophore composition is actually suboptimal for steady state ATP turnover. So this oval here indicates the stoichiometry of the chromatophore that I have shown earlier. And as you see here, you basically get a better turnover rate for this low light condition if you were to have fewer reaction centers and more BC1 complexes. The caveat for that, however, would be that you would not be able to respond to fluctuations in light conditions as effectively. And also, you would be ignoring the problems arising from damage due to potential overacidification of the membrane. So how do you climb from the scale of a single chromatophore to the cell? This is a collaborative work, again, with Neil Hunter's lab, where they provide us with information in terms of basically the count of chromatophores for a given growth condition for a Rhodobacter spheroides cell, because there's no way you can produce such an information by a first principles computation. You have to borrow it from experiment. And so my aim, as I go from a single chromatophore to the scale of an entire cell, is account for total ATP production rate integrated over the entire cell. Then ask the total expenditure of ATP for one cell as it makes a copy of itself. Subtract from this the base metabolism, which is not computed from first principles. The chlorophyll production channel alone has about 3 dozen steps, and it is impossible by any current technology to try to simulate this from first principles. Instead, the ATP cost of maintaining the cell per unit time is taken from experiment. And this is the only reason why such an estimate is possible in the first place. But with these bits of information, you can compute the time it takes for an entire cell to produce enough ATP to make a copy of itself. That is in effect related to the return on investment in terms of ATP for the entire cell. And of course, you know exactly what this is related to. This is related to the growth rate, because the doubling time for a cell is precisely the time it takes to generate enough ATP to make another cell if, and only if the following things are true. If I account for the base metabolism of the cell, if the soup within which the cell swims does not have an energy source, so my only energy source is sunlight, and if I am not permitted to have an energy expenditure channel such as motility, so that I expand energy only to maintain the cell and make a copy of it. The last two conditions are basically experimental control on the part of my colleagues at Neil Hunter Lab. And so there's a set of growth experiments that follow these conditions that was performed by Andrew Hitchcock. And you see already a very interesting behavior here. The growth limits itself past a certain light intensity. So Rhodobacter spheroides doesn't like a whole lot of bright light. It just doesn't do any better if you shine more light on it. And so can one reproduce this behavior from such a first principles computation? That is my aim. And this is my first major result. The doubling time here is compared between the experimental observations from Andy in Neil's lab and the computation that I have just outlined for you. That is basically a tally of the ATP production rate as a function of the illumination condition. And what you see is a quasi-quantitative match between the two, which fails to be accurate at extreme low light and extreme high light, which is reasonable because this model is actually tuned for the operational parameters, which correspond to something like 1 to 5% of full sunlight intensity. And the structural models that I have presented to you earlier are also for low light grown chromatophores. So it's probably fair that there are quality limits to this approach if you try to apply it over 4 orders of magnitude of illumination conditions. The second outcome that you get as a bonus from this approach is the energy return on investment as a device, either for a single isolated chromatophore or for an entire cell. So as I turn on light, more ATP is synthesized, but I have to also pay for the cost of a disproportionate amount of waste as my efficiency drops. So if I look at an entire cell, I see that my maximal energy return on investment actually sits about 3 to 5% of full sunlight intensity. This is not put by hand into the computation. This follows from the structural and functional description, and incidentally, it corresponds to where these bacteria actually prefer to live. So there's a quality control then of the model that I have presented. As a bonus, as an additional bonus, you get to compare a bacterium to human-made devices with similar purpose. So the energy return on investment for a photovoltaics device, let's say you pay $10,000 to put something on your roof. It will pay for itself in about a decade, and it will probably have a warranty about 2 to 3 decades, right? Do these sound about reasonable? So these are typically the numbers that you think about when you're trying to purchase a human-designed device. Of course, such questions of return on investments are usually asked in terms of currency, such as dollars. If you do the math in terms of the energy costs, these are the typical rates of energy return. For a barrel of oil, you can usually use the energy to dig up about 20 barrels of oil. 10 would have been acceptable, 40 would have been excellent, but there is almost no oil reserve left on Earth that is so easily accessible. And this is where a bacterium is. It clocks at about upwards of 40 in terms of energy return on investment. One caveat in this comparison, and this is very important. When you're comparing a bacterium to a photovoltaic cell, that number, energy return on investment for a photovoltaic cell, does not include the cost of the factory. So that number that is quoted here, 19 to 38, is not the energy return on investment of a factory that makes photovoltaic cells and is operated by the energy of photovoltaic cells. So this is unfair against the bacteria as an energy comparison. I emphasize this because in this slide is the rationale of somebody walking in and asking, why do you study energy harvesting in bacteria? You study energy harvesting in bacteria because they outperform anything that is built so far by humans in terms of, as quantified by energy return on investment. So normally this would be the end of my talk. Where do I go from here? Well, I'd like to repeat this construction for other systems, such as cyanobacteria and plant guarana, and eventually towards the mitochondrial respiration. And I would like to use energy return on investment as a comparison between the species. If one of the species can outperform the other and can out-evolve it, how would you quantify evolutionary fitness in terms of energy return on investment? And ultimately, if you can produce these rather than let evolution let us course over billions of years, this is essentially a rational design criterion. Because any bioengineered energy solution has to relate protein level changes to cell level growth efficiency. And energy return on investment provides a natural quantitative measure for such fitness. So this would have been the end of my talk, except, well, okay, can you explain it to the kids? First of all, why would you even want to? Okay, so when nobody is looking, I moonlight as a science outreach advocate. Don't tell anyone. But turns out that people have been interested in this problem pretty much as long as humans had a history. I mean, so this is basically the sun god Ra giving rays of life that are captured by the goddess of fertility, Osiris. Of course, from Bhagavad Gita to every Abrahamic religion, there's some description of the interplay between light and life. And the connection between vision and photosynthesis, humanity knew about it 2.5 millennia ago. So quantum biology is something we have been fascinated about for as long as we've been around. But forget poetics. This matters today in terms of the practical quality of life because energy excess essentially relates to prosperity across the world. And so humanity, of course, uses a vast amount of energy, but it's still just a piffle compared to what the sun provides to Earth. And ultimately, your quality of life is determined by free energy excess. So for something like quantum efficiency and excitation lifetime, there are ways to very accurately test these things, and then your room for wiggle is actually very small. And we have been able to reproduce this, meaning essentially the picosecond scale kinetics with great accuracy. Down here, you pay the price of your loss of detail as you climb the scales. So in that regard, it is already surprising that the low light turnover behavior where beyond 3 to 5% of full sunlight illumination, you lose reproductive advantage. That this follows from the computational model alone is almost surprising. And it is not put in by hand.
I guess I had a question, just a naive question, which is that, I mean, is it always the case that the rate limiting step for division is metabolic production of ATP as opposed to DNA replication or some of the many other things that have to go on for cell?
This is highly dependent on details of growth conditions. The statement I made in terms of the cytochrome BC1 complex being the rate limiting component is only true for typical physiological growth conditions which lack a number of strains. So for example, if you change the salinity of the environment, such a statement shifts drastically. And for example, cytochrome C2, which is not a rate-limiting component, suddenly becomes rate-limiting if you change its excess pattern by changing the electrostatic environment. So everything is conditional, and I emphasize again that the only reason such a comparison was possible at all was we were met more than halfway by our experimental collaborators that simplified the problem for us. Because otherwise, I'd be selling you a magic bullet that says, I can simulate a cell. That is not possible because there is still a 12 orders of magnitude complexity cost you have to pay, which cannot be done in principle generally.
So just a short answer to your question is that anybody who studies metabolism, they always give things that are called like GAMS, growth-associated maintenance, and non-growth associated maintenance. And they always relate back the growth conditions to the ATP turnover. Technically, it should be also ATP and GTP, but it is the dominant nucleotide there. So yes, that is always used as a metric. And yes, it can be sensitive to other environmental conditions, but that's all rolled into what they call these biomass equations. Torsten?
So you have these many knobs you can turn to get a different return on investment. Like which one matters most, or do they all matter a little bit? Or so if I wanted to get, or if I mess something up here, and what part is it where you see the biggest breakdown and so on?
Two separate answers to this. One, you can actually break the system, which the computational model cannot account for, because for example, if you change the cytochrome BC1 stoichiometry, you can actually damage the membrane, and such a mechanism is not accounted for anywhere in the model. But if I simply ask, what are my optimization criteria? I could give you a separate answer for steady state kinetics, in which case I would lower the number of BC1 complexes, lower the reaction centers, increase the BC1 complexes slightly, and add LH2 complexes to taste depending on what light intensity I'm operating at. Functionally, this is not how these species operate. They seem to favor robustness concerns over optimization with respect to ideal growth conditions. So they are all suboptimal, stoichiometry-wise, as if to prepare themselves for a hard day.
Yeah, I wonder how much you've made a huge multi-scale model, but I know from the area of not photosynthetic bacteria, but other bacteria, do flux balance analysis.
That's the right language, yeah.
Does this kind of thing, and essentially you measure the uptake rate of the substrate, and you determine the growth rate by how fast that can be converted to biomass.
Right.
It's a much smaller model than what you're talking about.
Right.
What I'm wondering, I guess, is your fitting essentially based on a flux balance analysis?
Not specifically in that language, but it is akin in spirit to that. I did not specifically use flux balance analysis, but in terms of the computational complexity and in terms of what is the ingredient of the model and what is the output, it is actually very similar.
Okay, well, I think we sort of exhausted all our energy supplies. We have to, our return on investment of energy is probably not as large as the bacteria. So with that, thank you very much. And I guess you're going to discuss